So welcome to the Bio 1-2 ATAR Notes lecture for January, so a Headstart lecture. Welcome to everyone. Hopefully this is the first day of lectures. You may have been to some already today. If not, again, welcome. So I'm presenting the Bio 1-2 for Tute Smart. And so again, we'll just be running through a lecture today. It'll go until 6:30 and it's about all things Bio 1-2, just especially Unit 1. And it's just going to give you a bit of a head start intro into what this subject kind of may include. So you may have been to some ATAR Notes lectures before. It does depend. I'm assuming most of you would be in year 11. Some of you may even be in year 10. Again, especially if you are in year 11, kind of going into VCE and maybe having a few other 1-2 subjects, maybe some 3-4s as well. ATAR Notes is a resource that you guys can use. So there's the little link in the corner there, and there's a bunch of high school kind of resources that you can use, and obviously everything catered towards VCE as well. There are a lot of different resources that I'll talk about in a second, but a lot of them are made by people who have been in your position before, so those who have gone through Bio 1-2. So it just makes them kind of really useful, relevant resources. So some of those things that I was kind of mentioning about, so there are some study notes. Some of you may be familiar with the ATAR Notes forum. There's also a couple of discussions, the lectures obviously that you're attending today, and just a bunch of other stuff as well that does become a little bit more relevant once you're kind of entering those VCE years. But then again, if you are in year 10, there are a couple of things there as well that may interest you. And again, these are all free, so you just need to register, again, at that link in the corner, and you'll be able to access them, and there's heaps of things there. And again, especially with bio, I'll talk about this as we go through more of the lecture. Bio is a really big and broad subject, but the thing about that means like it's studied by a lot of people around the world. Really, it's a really popular subject, so there is no shortage of resources, and there's heaps on the ATAR Notes website that you can have a look at. If you are wanting something a little bit more, we do have some other resources. So that does include our tutoring program, so Tute Smart. So for example, that's where I tutor bio. We also have some study guides and this program called Ed Unlimited. So I will talk about these a little bit more later in the lecture, but just kind of as an overview, TuteSmart is, we do kind of online tutoring and you receive them from tutors, again, like myself, who have all been in your shoes and in similar positions recently. Study guides, in terms of you guys may have seen them before, they're just sort of the books that may cover topic tests. They cover study guides that just cover the curriculum, and Ed Unlimited, which is basically kind of the online version of those study guides. But I'll get into those a little bit later and mention some of the kind of offers we've got going with them as well. But otherwise, I will head in straight to our lecture. So again, welcome everyone. So we're doing Bio 1-2 this year, and be aware this is the second year of the new study design. So there are kind of, I guess, at least you guys aren't starting off, being thrown straight into a deep end. There is a year that has gone before you with this new study design, but just be aware of that, especially with 1-2, because you don't get the exams, I guess, like made by VCA. Be aware when you're looking at resources that there may be some different ones from other kind of areas. So just kind of make sure that you are studying the right study design. In terms of some housekeeping, so you will be able to access these slides. There should be kind of a region below where you may be able to download them. If you aren't able to download them yet, you will get them after this kind of lecture. In terms of the Q&A, we have the Slido program. So you should be able to see that down below as well. And there'll be a little section there where you can type in any questions. I will kind of mention, we'll go through Q&A kind of at the end of each content block. So the way we'll divide it is kind of, as you can see there in the bottom, we'll do one content block kind of focusing on cells and things like that. And then we'll go into our second content block, which is a little bit more on systems. So in between, we'll have a couple of Slido polls where you guys can vote, and I'll just be testing you and see how well you're listening a little bit. And as well, you can pop in questions. As I go through the lectures, please feel free. Pop in a question anytime it comes to your head. I won't cover them like straight away as you're writing them, but at the end of each content block, we'll go through that Q&A. So kind of just before our break and just before the end of the lecture as well. Again, you can upvote any questions. So if you see someone's asked a question that you're wanting to ask, feel free to just like it, and then it'll come up a bit higher rather than just writing the same question. But yeah, please, by all means, go ahead and write, and don't be Kind of like, if you've written your question a little bit earlier on and I haven't gotten to it yet, just remember we'll get to them at the end of the content block. So please, any questions that come into your head while we're going through the content, feel free to pop them down. Okay, hopefully that will make sense to everyone. So the things that we're going to kind of cover, so this is Area of Study 1 and Area of Study 2 for Unit 1, so that's what we're going to be looking at mostly today. So Area of Study 1, I will say compared to the old study design, it's not too different. There are some kind of new things, but I would say things mostly got cut out rather than added, which again is kind of good for you guys. So we will be covering sort of like your basic biology stuff. So we've got our cells, our organelles. So we're thinking really almost on like a minute microscopic level. And then in our second half, we'll look at Area of Study 2. So this is when we're kind of zooming out a little bit and we're looking at things in terms of systems. So we're looking at the vascular system, we're looking at digestive, endocrine, some other stuff as well, and homeostasis. I think most schools do cover homeostasis in like junior science. So you may have been introduced to it a little bit like in, if not year 9, then maybe year 10. But yeah, we'll be covering that and looking at that a little bit more closely. And again, just remember we've got about a 15-minute break halfway through and we'll cover Q&A as well. So hopefully that is kind of making sense. Okay, in terms of before we jump in terms of who I am, I don't think I introduced my name at the start. Sorry, well, my name is Loz. I am a bio tutor. I graduated in 2020, so I'm going into my 3rd year of uni now. So I'm doing med at Monash. So I did bio 1, 2 and bio 3, 4, just kind of the typical one. So I did 1, 2 in year 11 and 3, 4 in year 12. And so I got a 45. I really enjoyed bio 3, 4 and I enjoyed bio 1, 2 as well. I know there's usually a lot of questions. I'm sure we might get some later this afternoon, or I suppose, I don't know if you guys are already thinking about year 12, but in terms of how relevant 1, 2 is to 3, 4, that's a really common question that comes up. For me, and I think what I assume, you guys after going through 1, 2 and assuming you do continue on into 3, 4, it just gives you a little bit of confidence because you do cover quite similar principles in 3, 4. And it's not to say that you can't do 3, 4 without 1, 2, obviously not. There's a lot of students that do that. However, it just does add a little bit of confidence. And I think it essentially does reduce your learning a little bit because you've got less to cover if you've already looked at some of the stuff that we're about to look at now and some of the other stuff that you cover in Unit 2. That does translate to Units 3 and 4 as well. Okay, so before we jump in, we'll start off with the little Slido poll, make sure everyone kind of knows how it's working as well. This is just a really general open-ended question. So it's about what you're most concerned about going into bio this year. So I'll share the poll in a second. But for this, I am really mostly referring to, let me make sure, yeah. I know like you guys are only starting perhaps year 11 or maybe year 10. Hopefully there's not too many things that you are concerned about. So I'm mainly just asking here, like, if there's anything that you think may be tricky going into bio, or if you have any, like, not reservations about the year ahead, but what you think you might struggle with a little bit, or perhaps, again, this is like the first 1, 2 that you're doing, maybe different approaches you think might be necessary, especially from that transition from your like general year 10 science into your VCE, like your units 1, 2. Yeah, so hopefully you guys don't have too many concerns, especially it is only the start of the year. Yeah, good to see a couple of things coming through. We'll go through them. Pedigrees and cell cycle, yeah, so in terms of like actual content. So we'll cover the cell cycle today. Pedigrees we won't, but that would come in later on in the year. But yeah, cell cycle and some of the content as well. Some of the content will be fairly, I don't want to say easy, but like easier to grasp, especially, sorry, things like cells and stuff like that that you might have covered before. However, yeah, going into new stuff and stuff that might be a little bit more technical, that can be tricky. So that's definitely something that we'll look at today. Yeah, so a lot of stuff on content. What I was going to say as well is I really enjoyed 1, 2 and kind of doing the 1, 2 and not going straight into the 3, 4, because for me, it gave me a little bit of an idea of like trial and error in 1, 2. And I always say this, even with some of your other subjects as well, like some of the other 1, 2s you guys may be doing, use this as a bit of a trial year to figure out what works for you, what doesn't, because some of the subjects are really quite specific, especially bio. Bio in year 12, there's a very specific way of marking, there's a very specific way of answering questions, and kind of mastering those skills as best as you can in year 11 is a really, really good thing to try to do. So hopefully that makes sense. I'll go through. I wonder if I can. Okay. So I can see, yeah, talking about workload. Learning how to memorize important stuff needed for exams without needing a paper copy of my notes. Yes, that may be a new thing as well, depending on your guys, like on the schools that you go to. I don't want to say they spoon feed you, but sometimes, yeah, you may be used to having notes, you may be used to doing like open book exams, stuff like that. That will be a little bit different this year, and obviously as a practice for your exams next year. So yeah, kind of, I guess year 11, or again, 1, 2 is where you start to take exams quite seriously, especially because they are essentially that warm up for the big exam that will be 3, 4. And remembering with 1, 2, I'm trying to think. I think most schools will do it that you will have your mid-year exam. So you'll have an exam in June, and then you'll have your exam in November as well. Remember that in year 12, you just have that one exam. So get kind of used to this idea of being assessed on a whole year's of work as well. So it being a lot to remember. I'm not going to lie, bio is a relatively content-heavy subject. Again, with this new study design, I would say more stuff was taken out than was added. So 1, 2, we'll go through what's in Unit 1, obviously, in our lecture, and you guys will get a bit of a taste to see the content. There is content, I'm like not going to lie, there is a lot to remember. But I think it definitely is doable, especially if you learn what works for you and what doesn't in terms of getting stuff memorized. So yeah, it's a lot of the same stuff, memorizing the content. Yeah, being overwhelmed with the workload. Again, with that, try to stay on top of it as best as you can. This is really vital for year 12. So it's super important if you can try to, again, hone the skills in year 11. If you fall behind, think about, why did I fall behind? How can I not do this again next year? And how can I now catch up? Staying on top of the workload is really, really important. And I really encourage you guys to kind of always be prioritizing that. I know a lot of people may think about getting ahead on the content, and like it's up to you, it does depend. That can work. And again, because there is quite a bit of content that may be helpful. However, yeah, it is up to the individual, but just try to always stay at least up to date to not fall back. So yeah, learning the content, extensive vocabulary. Yep. So kind of learning, it might be good for me to mention now, with biology, I highly recommend keeping a glossary. So again, super important for year 12. If you can get into the habit of it in year 11, it'll help you a lot. Because a lot of the questions are definition based, and that can be a little bit different from other subjects, obviously, and even other sciences. Bio is very wordy, you know, compared to some other sciences and stuff that may be a little bit more formulaic. So a glossary, I really highly suggest that. Retaining all of the information. Yeah, of course. Yeah. Okay, so it looks like you guys are kind of sharing the same concerns in terms of a lot of content. Yeah, not just memorizing it. This is a really important point, not just rote learning stuff, but understanding it. It will help you so much. Try your best to not just copy paste like a robot. Try to actually understand the concept. I think I saw something in here about learning how the concepts relate to each other. That can help a lot, especially into 3, 4 as well. A lot of the subjects on the study design or the concepts are intertwined. So if you can remember how one leads to another, and I always emphasize to my students, bigger picture. Always think of the bigger picture to help you kind of understand and put things into perspective. And that just helps me a lot with learning. Yeah, we'll talk about study tips. I think, it's already been 15 minutes. I think I'll keep going for now, but I'll definitely try to address these as best as I can. So kind of questions, yep, new words and definitions. And obviously don't beat yourself up a lot as well. Like, it takes time. Don't expect this stuff to come really instantly. It's really good that you're, coming to this lecture because you'll get a bit of an introduction. And then once you learn it in class, again, that's more time to consolidate. It's really important as well. Just always be consolidating your knowledge. Try not to learn it. And again, this is good practice for year 12. Try not to learn it and then kind of, pass your little test and then throw it away. And then focus on whatever else. Try to, again, when you learn your content, like the stuff we're going to go through first, like cells and cell cycle, learn that and then try to revise it throughout the year. So then, if your school does a big kind of year-long exam, you're always keeping that knowledge up. And again, it's mimicking year 12 because I think a big thing that can help, that can ultimately lead students to falling behind is when you have to kind of relearn information. So try your best to kind of, once you've learnt it, revisit it. And I'm not saying like revisit it every day, but like, maybe every month go back to it, or every 2 weeks or something like that. Just so you're not learning something in February and then by, October, or even like if you have mid-year exams, like by May, you're going to have to relearn everything that you've forgotten. So that is a good thing as well. Okay, yeah, so in terms of practice exams and stuff like that, I'll talk about that a little bit later. I think I'll answer some of the Q&A a little bit later on. But yeah, so I can see most of you guys share kind of the same concerns. And those are the concerns that I'd expect you guys to kind of have as well, because again, this is a bit of a transition, kind of a new thing going into a 1 and 2 subject, especially bio, which can be a little bit tricky. Okay, so again, I'll try to answer these questions as best as I can. We'll do our Q&A at the end of this content block and at the end of the lecture as well, and we'll kind of come back to some of those things that you guys were talking about. If anything pops up regarding specific questions, again, pop it in the Q&A. And if anything comes up regarding content that I go through, if you need me to explain it a little bit more, or you're kind of missing something, pop it in the Q&A and we'll cover it. So to start off, again, we're kind of easing in a little bit nicer here. We're starting off with cell structure and function. So we're looking at prokaryotes and eukaryotes. Again, these terms should be relatively familiar. It does depend on your school and obviously what you guys do learn in junior science. But generally things that I've highlighted here, like you'll kind of know if these are a big term. And I would start now making your glossary. And as you learn stuff in class, make that glossary and add to it throughout the year. So when you come to exam time and when you're learning how to do your practice questions, those definitions for these keywords, such as prokaryotes, eukaryotes, it's really clear in your mind. Okay, so in terms of some differences, so we can see our prokaryotes on the left and our eukaryotes on the right. Generally, and I want to say generally, because don't think of it strictly like this, but I like to use examples when I think of things in my head. So often I think of bacteria as our typical example of a prokaryote, and like an animal cell, for example, as our typical example for a eukaryote. Bearing in mind, there are more prokaryotes than just bacteria cells, and there are more eukaryotic cells than just animal cells. But for me, that's how I would kind of like remember it in my head. Like instead of thinking, what's the difference between a prokaryote and a eukaryote? I used to think in my head, what's the difference between a bacteria cell and an animal cell? But obviously, think about exceptions and stuff like that. Okay, so no membrane-bound nucleus in a prokaryote, and in a eukaryote there is, so that's what we can see kind of here. The general rule of thumb with these two is prokaryotes are very simple, eukaryotes are a lot more complex. So no organelles, we have membrane-bound organelles. We'll talk about organelles in a bit more detail in a second. But yeah, again, really simple, doesn't really need the technical stuff that organelles do, whereas eukaryotes do require it. Not capable of true multicellularity. So we technically think of prokaryotes, definitely in the context of Bio 1-2 at least, as this idea of single-celled organisms. Eukaryotes, we tend to think like a little bit more as multicellular, but be aware there are single-cell eukaryotes. So kind of maybe put this as like a difference that's a bit lower down your list in terms of ones that jump to your mind. Prokaryotes, we'll talk about binary fission a bit later on, but they reproduce by this like really simple method of dividing, whereas eukaryotes, we're thinking about mitosis, and for this actual idea of reproduction, we're thinking of meiosis as well, but we'll talk about that a little bit later. So as you can see, there's some kind of differences, and you can even see like visually as well, that idea of the nucleus is a big one, the organelles are a big one as well. Okay, so that's the stuff we can kind of see there. So organelles in general, here we have an animal cell. We've got a couple of organelles here. Obviously, you can tell by the way they're drawn. We've got like our endoplasmic reticulum and our Golgi, our nucleus. Some of the other ones are a bit more ambiguous. Basically, with your organelles, know what I don't want to say popular, but the major ones are, especially in like an animal cell or a eukaryotic cell. Know their general functions. You don't have to be too specific. Again, your teachers will emphasize which organelles you need to know heavily. On the study design, they emphasize chloroplasts and mitochondria. So those are 1000% you need to know them. But yeah, be aware there are some others where you just need to know their functions a bit more generally. and kind of what they may generally look like. Whereas, for example, chloroplasts and mitochondria, we look at them a lot more closely. But yeah, just generally know some of their functions, and know also what they will look like on a micrograph. This is a skill obviously needed in 1, 2, and 3, 4. So here we have kind of like our typical drawing of a mitochondria, and you'll know what they look like in a diagram as well, but understand what they will look like on a micrograph. When I say micrograph, like a picture of a cell taken with a microscope. So usually they're like, if you go through a couple of year 12 exams, you'll see kind of what I'm talking about. So they'll be like black and white, they'll be grainy, and they won't be this kind of clear, nice, color-coded, really distinct kind of organelle that you see like on images like this. So just make sure that you're looking at key features, because you may be asked in an exam to like label a micrograph. And if you're used to seeing mitochondria as this typical orange squiggle, and then you're seeing this in black and white and it looks a little bit different, then yeah, you may get a little bit confused. So kind of understand what it looks like, I guess, in real life as well. Okay, so here is a general table. Again, I'm not going to describe them in a lot of detail. Essentially, our lecture is 2.5 hours, so I will have to move through some things relatively quickly. Again, if you have any specific questions, pop them in the Q&A and I can come back if we have time. But yeah, because you guys are getting copies of these slides as well, just bear that in mind. So if we think of our really important ones, again, mitochondria and chloroplast, we'll talk about in a second. But we think about them, basically they are important and they're singled out on the study design because of their role in our biochemical pathways. So you guys would have likely heard of cellular respiration. So we think about that being associated with our mitochondria, and our photosynthesis being involved with our chloroplast. Bear in mind as well that the details of these have been removed from the study design. You do need to know them in year 12 though. They are really important. So I guess there's no harm in learning about them, but just be aware that you shouldn't be tested on them in such detail as you may have been previously based on the previous study design. Okay, hopefully that makes sense. So nucleus, really important. We're thinking about the house of our DNA, so our genetic information. Ribosomes, we're thinking really important for protein production. Again, you'll learn about the really nitty-gritty details of protein production a little bit next year. So our endoplasmic reticulums, we're kind of thinking here like packaging and transport. So you can see here, it's got like modification as well of our proteins. So think about that. Again, rather than rote memorizing, rough endoplasmic reticulum does this, smooth endoplasmic reticulum does this. Think about like, what's the difference between the rough and the smooth? The rough has ribosomes. What do ribosomes do? Protein. So therefore, what does the rough endoplasmic reticulum do? Packaging, modifying proteins. You can see that kind of train of thought rather than thinking like everything separately, thinking like ribosome, proteins, smooth endoplasmic reticulum, steroid molecules, rough endoplasmic reticulum, proteins. Like think about it, I guess like think smarter, not harder, maybe like try to tie concepts together, tie organelles together. And then like obviously what I've done is a little bit simple in terms of just thinking rough with ribosomes, but that kind of thinking or logic can be applied to so many other concepts in Bio 1, 2. Okay, so with that, smooth endoplasmic reticulum, again, smooth because no ribosomes, therefore not associated with our protein transport, instead with our steroid molecules. So you don't really talk about the smooth endoplasmic reticulum a lot. Like that's kind of just enough to know that it's just like synthesis of what you might call like your lipids. So just your steroids and other stuff. You like never talk about it, really. I don't think I ever wrote anything about the smooth endoplasmic reticulum in Bio 3, 4 or 1, 2. But yeah, so just know it generally. Golgi, again, we're thinking of protein. So it's basically the next step after your rough ER. So thinking about further modification, and this is more so like the actual movement and packaging of the protein ready to be sent out of the cell. Lysosomes are thinking of basically our like rubbish bins. So your unwanted stuff, your useless stuff, I guess, in the cell gets digested there. And then again, we've just discussed our mitochondria and chloroplast as well. Again, there are more organelles that may be in your textbooks and you may learn about in class that aren't covered here, but these are just some of the general important ones. Okay, so zooming in a little bit on, here's mitochondria, here's our chloroplast. Bear in mind this diagram is overly detailed. You don't need to know about like porin, we never talk about that. But just generally, again, know what it looks like. Maybe be aware of the different parts. So something important with mitochondria and chloroplasts is they are double membraned, as you can see here. So they've got two membranes. And so you can see this is the outer membrane. And like obviously this is quite clear where the two membranes are. This squiggly thing is the inner membrane. So just be aware of that. Don't think of it as like something plopped in a shell. Like this is one membrane and all this squiggly stuff is actually the other membrane. So that's something important to remember. They are double membraned. Maybe remember some of the terms. Again, in terms of the locations, it's important for the process of cellular respiration. Same with chloroplasts for photosynthesis, but we don't talk about that in detail a lot now. Next year, it's something that becomes a bit more relevant. But basically, important terms to remember should be matrix and cristae, really. So the cristae is this inner membrane. It's just kind of another name for these folds, especially. And then the matrix is just the fluid that kind of flows inside here. Okay, in terms of the chloroplast, we're thinking about this obviously with photosynthesis, so in plants. So we have these kind of stacks. Again, I'm not going to go into a lot of detail about what they do, but we can see they always compare them to pancakes. They look like a stack of pancakes. So there's like a bunch of different names for them, but basically I just call them like thylakoid discs. A bunch of thylakoid discs together is a granum. So 1 granum, 2 grana. So again, it's like, you'll get the hang of it with the names and all that. But yeah, so think of the grana and basically the stroma. So the stroma is kind of the chloroplast version of the matrix. So the matrix is just the fluid. That's kind of the same thing. The stroma is just the fluid. You can almost think about it as the organelle version of the like cytosol, I suppose, to kind of draw a bit of comparison. But yeah, again, the names of these, the reason why I'm flagging these is because they are important for different stages. So a certain stage happens in the stroma, a certain stage happens in the grana, certain stage of cellular respiration happens in the cristae, in the matrix, etc. So hopefully that kind of helps you out. Again, when you, if you like download these slides and look at them, just be aware that they are fairly detailed, like lipid globule, starch granule, like you don't need to know that stuff. Just be aware of the kind of key things that I've mentioned. Okay, a Slido poll on our organelles now. So what organelle is not involved in protein production / transport? So we've just got a couple of polls kind of scattered throughout the lecture. They're hopefully not too intense in terms of content to remember. So what organelle is not involved in protein production? Looking good so far. I think there is a little bit of a lag between what I'm saying and then once it reaches your devices. So I'll give it like 5 more seconds. Okay, looking pretty good. Perfect. I might stop that there. So the answer is the smooth endoplasmic reticulum. So with our ribosome, we're thinking of our protein production, so where the protein is actually made. Then moves on to our rough endoplasmic reticulum for further modification and transport, and then into our Golgi body, where we've got further packaging there. So remember, the smooth endoplasmic reticulum is more so associated with our steroid molecules or our lipids, kind of things like that. Okay, so moving on to a little bit of stuff more so in the cell. This is probably the most chemistry-ish it gets, or like scientific, I mean, obviously bio is science, but you know what I mean, like that sort of stuff. So we think of things being hydrophilic or hydrophobic. So again, these would be key glossary words. So philic means to like, hydro refers to water, phobic kind of means dislike. I think of it as like, phobic, afraid. So just think of that, like hydrophilic, like kind of going towards water, liking water, hydrophobic, almost like repelling water. So that's essentially what we think. If something is hydrophilic, it likes water, it will dissolve in water. So you can kind of think of sugar, when you stir sugar in water, that dissolves. So you'll also find that we refer to that as polar. If you do Chem 1-2 or maybe Chem 3-4, I don't know if you're doing a 3-4, this will be a really common word that comes up. Lipophobic, yeah, I never really use these terms. I would either use hydrophilic or polar. And then so the opposite of that is hydrophobic, so repelling water. So again, think of like when you stir sugar in water, that dissolves. When you stir oil in water, that separates. So hydrophobic repels water. And so we can call that non-polar. And the thing here is that hydrophilic or polar molecules will mix with polar molecules. And hydrophobic or non-polar molecules, as we see here, they mix with their other hydrophobic molecules. So they're very separate. They only like to mix with the same type of molecules, if that sort of makes sense. So yeah, here's kind of a visual depiction of what we were saying. No matter how much you try to force them to interact, there's that separation. Whereas if this was water with a hydrophilic molecule, you'd be able to shake it up and then you wouldn't see a separation because it would be mixed in. Okay, the reason why we talk about this kind of stuff is because we look at it in the context of the plasma membrane. So you can see this is meant to be a cell. So the plasma membrane, what we were talking about before, in terms of how, like chloroplast and mitochondria have a double membrane. So the plasma membrane just refers to the membrane that wraps in the cell, essentially. So it looks like this. So you can see that it is 2 layers, which we'll talk about. And we often refer to it as this fluid mosaic model. Again, that's something that may come up. I probably won't go into it in detail here, but it's just this idea that the plasma membrane allows stuff to pass through it. And it's also got, as you can see here, it's quite complex. It's got other bits in it as well. So we talk about the plasma membrane in the context of transport. So we can see here, as I mentioned, the 2 layers we've got. So there are phospholipids. Don't really worry about the word like amphipathic. I never used it. Just think about this idea that they have both the 2 words we talked about before, hydrophilic and hydrophobic. They're the 2 components mixed into one, which I know I said wasn't a thing. So that's why one end is hydrophobic and one end is hydrophilic, as you can see here. So the head, it will mix with water. The tail does not like water. So that's why we get this kind of formation like this. The tails gravitate towards each other and they're on the inside of the plasma membrane, and the heads will face the outside because on the outside will be water, will be polar molecules, right? So then that's why the phospholipids basically orient themselves in this way, and then by doing that, they form this plasma membrane essentially. So that's why you'll often hear the phrase like double layer phospholipid wall or something like that. Phospholipid bilayer is probably a nicer way to put it. And so that's what basically makes up the plasma membrane. So for a molecule to pass through a plasma membrane, so basically from inside the cell to outside, or the reverse way, they have to get through this phospholipid bilayer. That's just a bit more of an in detail drawing of that. So in terms of things that are embedded throughout this plasma membrane, we have cholesterol. So I suppose like these little yellow things are resembling our cholesterol. So these are important basically in maintaining fluidity. So they don't allow these molecules to get too close, but they also don't allow them to get too far. That is the general principle of it. So it's really important in making sure that essentially the plasma membrane is fluid enough, but not too loose. Hopefully that kind of makes sense. Also got this idea, of forming a glue. So you can see here, preventing them from becoming too rigid, preventing them from coming too loose. Okay, so here is where I guess like the money is. We're thinking about transport across the plasma membrane or across membranes in general. So the most simple kind of form of transport we can think about is diffusion. So this is basically when there's no like airs and graces about it really. You just have molecules coming from a region of like a high concentration and they just move to a low concentration. You'll often describe transport using these terms. So high concentration, low concentration, passive movement, net movement. And for me, I think like it felt like I was learning something foreign, but if you just break it down, like think, when you see here, like you've got something in a kind of clustered environment, it's just natural that it wants to spread out. So remember with diffusion, you don't necessarily have to think of it in the context of cells. Like if you think of perfume, when you spray perfume, when it comes out of the bottle, it's in that high concentration. The natural inclination is to diffuse. So it wants to reach out into like other corners of the room, where there's a low concentration of that perfume, if that sort of makes sense. So hopefully you can kind of understand it that way. And you can see with the diagram, it starts off, I know this is like really slow moving, but yeah, it starts off in this cluster, and it's not the natural inclination for the molecules to stay there. They want to separate. And the idea is they're going to spread out equally. So it's not that you're getting a cluster from here moving, like all of them moving to the bottom right corner. They're spreading out equally. It's that idea of high concentration to low concentration to achieve this hopefully like equal spread of molecules. When we refer to the word passive, it means it doesn't require energy. If you have something going down a concentration gradient, be familiar with these terms as well. Down a concentration gradient versus against. Think if you're going down, you're going from high to low, down a concentration gradient. That doesn't require energy because again, it's the natural pathway that the molecules will take. If you're wanting to move molecules from low to high, that's unnatural. That's going to require energy, but we'll talk about that a bit later. But that's just what passive means. It doesn't require energy. It's just what naturally happens, really. Okay, so we can see that there. Okay, so we also have facilitated diffusion. So when we refer to simple diffusion, it's basically what you think of when you think of like general diffusion. So in terms of the plasma membrane, it's molecules moving straight across that lipid bilayer. Remember, sorry to flip back and forth. The inside of the bilayer is hydrophobic. Yeah, the tails, these repel water, whereas the outside is hydrophilic. So just keep that in mind as we go through transport. So we can see here, things that travel by simple diffusion, hydrophobic substances. So this makes a lot of sense. You may think like, you know, the heads are hydrophilic. How do they get through? It's like the heads are only like kind of little, so they can just like weave their way through, really. But if you're thinking about like alcohol, steroids, these hydrophobic substances, yeah, they remember the tails. So that hydrophobic tail portion, they want to interact with them because they're the same. They're hydrophobic. So that's why they just scoot straight on through. You can have some molecules that may not necessarily be hydrophobic, such as water, but because they are very small, they can just squeeze through as well. Even though the tails like don't really like them, they can get through. And remember this idea of them being uncharged. So they're not positive molecules, they're not negative molecules. So they can still get through. In terms of facilitated diffusion, again, this is diffusion because it follows this general definition of it's passive, there's no energy required, and you're going from high to low, but it's facilitated because it requires a transport protein. So if you remember that image we looked at before, they have, you know, sometimes these protein channels, and they'll basically be like a little gateway to go through the bilayer without actually having to cross those phospholipid tails, because obviously they wouldn't allow them to. So by having these transport proteins, it allows our big molecules, our hydrophilic molecules, and our charged molecules, so ones that wouldn't be able to go through without the transport protein, it just allows them to scoot through, basically gives them a direct channel. So that's what we'd call facilitated diffusion. Again, it makes sense in the name. Simple diffusion, you just go through. Facilitated, you need something to facilitate the diffusion. Okay, the other one we look at is active transport. So this is what I was talking about, kind of the opposite of diffusion. We're going against that concentration gradient. So we're going from an area of low concentration to an area of high concentration. So because we're going against that concentration gradient, we need ATP. So basically you need this energy to like shove the molecule against what it naturally wants to do, if that's what it makes sense. You can kind of think of it as like, when you think of down a concentration gradient and against a concentration gradient, think of against, you're going up a hill, you need that energy to kind of push things. Whereas if you're just going down a concentration gradient, you roll the ball, it goes down. You don't need to, you know, constantly push it down the hill. That's kind of how I used to think about it. But yeah, so you can just see this here from a low region to a high region. Okay, in terms of osmosis, this is a very specific definition you have to be aware of. So net movement. You would have seen the word net in the other definitions as well. Basically, because if you remember, you can kind of see it in here, but like that little GIF, that little animated thing that we looked at with the red molecules dispersing through the black square, the molecules never stop moving. So they reach what we call equilibrium. We saw think about that perfume, like it'll get everywhere. And it's not like the molecules, once they've reached, equilibrium and once they're nice and separated, they don't just freeze. Molecules are always moving. Yeah, think about your general, physics principles. They're always moving. They never stop. So that's why it's called net movement, because ultimately, once you've reached that equilibrium, you will have some molecules that go back, if that makes sense, like back to that direction of high concentration. But obviously, if you have one molecule moving that way and 70 moving the other, it doesn't really make that much of a difference. So that's why it's net movement. So net just means overall. So hopefully that makes sense. So if we can see here, I'll explain osmosis in a second, but you can see that the movement is generally to the right. Some molecules will be moving to the left because that's just how they move. But again, it doesn't really matter because it's the net movement that we're looking at, the overall. Okay, so net movement of water molecules. So when we're talking about osmosis, specifically talking about water, so osmo, yeah, it means water. The other ones we're looking at, active transport, diffusion. Again, I could be talking about perfume. It doesn't really matter. With osmosis, we're talking specifically about water, and we're talking specifically across a semi-permeable membrane, and it's from a region of high concentration to low concentration. So with the other ones, again, like simple diffusion, in this context, we're thinking of it against a membrane, but it doesn't necessarily have to be. With osmosis, we are thinking of that semi-permeable membrane. So we think about the plasma membrane, basically. That's kind of what they're referring to here. So we're going from a region of high water concentration. So if you look at the left, there are more water molecules there. Like you can count them individually. There are more water molecules on the left than there are on the right. So the water is going to want to achieve equilibrium, and that's why we're getting water moving to the right. Hopefully that kind of makes sense. And that's why we see the water level changing. So the amount of water in the left decreases and the amount of water in the right increases, because we're moving towards that right side to try to equal the level of water, if that's what makes sense. So hopefully that is kind of okay. Okay, so in terms of osmosis and what direction things go, so you have your hypertonic, your isotonic, your hypotonic. So for this diagram, this is inside. So obviously this is your plasma membrane and this is outside. So if you were to describe a solution as hypertonic, basically there's more solute. So you think of think about the word. So tonicity is basically, again, I don't want to get too technical here. But tonicity, basically you can think of it as like how much solute is there. So in this case, we think of sugar. So you can describe sugar as like solute, basically. It's just something that's dissolved in water. So with our tonicity here, it's hypertonic because there's more solute in this area than here. So the outside of the cell is hypertonic compared to the inside, hyper, more, yeah? So what water is going to do is, if there's more solute outside, that means there's less water outside, right? So water is going to move from inside to outside of the cell. It's going to move to the hypertonic solution. And that's what we kind of see here. So this is hypertonic. Water is going to go to it to try to balance out the amount of water, right? To balance out the concentration. If something is isotonic, they're pretty equal. So outside, there's kind of the same amount of solute as inside. Remember, water will still move, but the net movement will kind of just be the same. So the amount of water going out is the same going in. Hypotonic, you're probably seeing where I'm going with this. It's the opposite. So there's more solute inside, or as I should describe it, hypo, lower. There's a lower amount of solute outside than there is inside the cell. Therefore, we're going to see water come in. What the application, I guess, for this is, if you place a cell, so we're thinking like this little yellow cell here, if you place it in a hypertonic solution like this, water is going to leave the cell and the cell is going to shrink. If you put it in a hypotonic solution, water is going to come into the cell and the cell is going to swell. So that's kind of the general application. Hopefully that makes sense. Okay, so this, I'm going to go through this fairly quickly, but it's about surface area to volume ratio. I know it looks like a lot of maths. You can kind of break it down when you have a look at the slides. But the reason why you talk about this is because the question you're meant to ask yourself is, why are cells so small? And the reason is the smaller the cell is, basically the higher the surface area to volume ratio is. So you can see with the bigger cube, you have a 2-to-1 ratio. So there's 2 times as much surface area, so the outside, as there is like what's inside. It gets a little bit smaller. Now you have 3 times as much surface area compared to what's inside. Smaller again, you have 6 times as much surface area compared to what's inside. So the smaller a cell is, the higher the surface area to volume ratio is. And that's beneficial because think about the plasma membrane. Think about things coming in and out. You need nutrients, you need oxygen, you need stuff that's important to come in, and you need waste to come out. So basically the cell has to be very, very efficient at transport. It needs things in, it needs things out, and it needs it done properly, and it needs it done on time, right? So that's why cells are so small, because it means they have a huge surface area compared to the volume of the cell, so that the exchange of, nutrients, the exchange of molecules is maximized. I think a way my teacher used to say is, if you can think about like the nucleus or something, or think about a dot in the center of these cubes, think about how long it would take to get to the center of this cube, right? Based on the size. And you can see that the smaller you get, it's easier to get in because there's more surface area to that volume. Hopefully that kind of makes sense. I feel like this is something that may take a while to sort of grasp, but hopefully the general principle is kind of there. Again, be aware, I am going through these things fairly quickly. You will learn them again in class, obviously. And so don't expect, like, if you're not grasping it immediately, don't think like, oh, I'm so silly. Like, it takes time. It takes time, so don't stress too much. Okay, with saying that, we'll do another Slido poll. So, active transport involves the movement of molecules down or against a concentration gradient and with or without ATP. Mm-hmm. Okay, I am thinking about time, so I think I'll get through these polls a little bit quicker. But yeah, again, essentially that is what we're seeing. So against a concentration gradient. So active, we're going from an area of low concentration to high. So think of going up the hill. If we're going to push something uphill, we need energy. So it's with ATP. And that's where the active part comes from. So the opposite of passive, so without energy, is active. So bear that in mind. Okay. So hopefully that makes sense in terms of cell structure and function. Again, ask any questions. We'll get to it in a little bit. But for now, we're going on to the cell cycle. Again, this is a pretty detailed diagram. Most of the diagrams we've got are relatively detailed. So I suggest there's a bunch of good information, like come back after and look at these slides in a bit more detail. Some of them like even a little bit too detailed, like kinetochore, you don't really have to know that. Generally, I kind of hate this diagram. It's a bit confusing because it goes anti-clockwise. I don't know. I just like it when they go clockwise. But we have this idea of interphase, and then we have mitosis. So these little ones are like prophase, metaphase, anaphase, telophase. Those are steps within mitosis. So this whole thing is M phase, mitosis. What we're looking at here is interphase. So this is before the cell divides. So basically, in general, this eukaryotic cell cycle, it's about growing and then dividing and growing and then dividing. So relatively logical approach if you think about it. So we have our 3 main phases. So we have G1, we have our S phase, and we have G2. So I'll talk about these in a second as well. But G1 generally, like G, we're thinking of like growth, basically. That's how I used to remember it. So the cell is growing. S phase, the cell is synthesizing. So basically we're just doubling the DNA, because remember, we're a cell. We want to divide. We want to go into two. We have to double our DNA so we have enough DNA for another cell. If we didn't double our DNA, then like if we had 10 pieces of DNA to begin with, then we've got 5. Then we've got 2.5. It doesn't really work that way. So you have to double it to 20. So then you can divide into 10. Double it to 20. Then you can divide into 10. If that makes sense. So yeah, that's the general overview. So that's our interphase when we're working towards mitosis. And mitosis is the actual separation of the cell. Okay, so talking about mitosis, we have our little acronym PMAT. Again, you guys may be on top of this. You may have been taught it before. Remember that interphase is not part of mitosis. It's separate. So here is where we're getting like our growing, stuff like that. So you can see replication. We then move on to our P, so our prophase. And this is where our chromosomes start to become visible, basically. We can see we've got our replicated DNA. They're getting more condensed. They're basically getting like thicker, I guess you could say, because they need to be condensed so that we can, I guess, like move them around the cell a little bit more easy. So that's what we're thinking about in prophase. Then in metaphase, think of M for middle. So we're lining up along the middle of the cell, as we can see here. We have our little spindles. You can think of like spindle fibers, and those will attach to the middle of the chromosomes. So that's what we're thinking about in metaphase. Think about middle. We're lining up. And then that leads on very nicely to our anaphase, where we've lined them up nicely. We've attached our little like harnesses, I guess you can think, and then we're pulling them apart. So we're now separating our chromosomes, because remember, they're replicated. There's 2 of them. So now we're separating them. So one goes to one cell, one goes to the other. So we have the right genetic information in each cell. So we can see our little spindle fibers, they retract. I don't know, think of it like a bungee cord or something. So they're pulling the spindle fibers, and again, we're going to either side. And that makes sense because, again, that nicely leads us. Think about how it's leading. It's a process. It's a flow. So now that we've got them on either side of the cell, that's really great. We can just cut them in the middle, and then we've got 2. So we then have our telophase. So this is when we're now on separate sides of the cell. We now start to become enclosed in our little nuclear membrane. Remember that in general, like we talked about our nucleus before, it's our little house for our DNA. When we have our prophase, our metaphase, that kind of gets broken down, and our chromosomes just go wherever they want to go in the cell. Now we have to put them back in our neat little box. So we can see our nuclear membrane, as you can see here in yellow. This is reforming, so we're getting them popped in, separated nicely. Our chromosomes basically begin to decondense, so they kind of unravel. They don't have to be as tightly packed as they were. So then they basically become invisible again. And then you have interphase. So then each of these individual cells, understand that they are separated by this point. I know they look nice and conjoined, but they are separated. And then you've got your interphase, and that starts again. So then those 2 cells will continue to grow. They'll replicate their DNA, and then they'll divide. So hopefully that makes sense. So yeah, prophase, you just think of kind of the chromosomes appearing, the nuclear membrane disintegrating, I guess you could say, or breaking down. Metaphase, think lining up in the middle. Anaphase, think of apart. Telophase, we're kind of getting that nucleus reformed. Remember, there is an important step of cytokinesis, and that's the actual separation. Cytokinesis is not a part of mitosis. So as you can see, we'll talk about it in a second. But as you can see here, telophase and cytokinesis. So cytokinesis is the actual separation. But yeah, so we think of PMAT as those 4 major steps of mitosis. So here is cytokinesis. So this is what I'm kind of talking about here. So basically, you don't have to worry too much about terms like cleavage furrow and things like that. But basically, now we've got our 2 like mini nuclei. We just basically pinch off that middle bit. So then now we're separated into 2 cells, as you can see here. So it's just kind of what you expect would happen. In plants, it is very similar. However, because there is a cell wall, remember in animal cells, we don't have cell walls. In plant cells, they have cell walls. So that's something that needs to be accounted for as well. So everything is the same. You've got your PMAT that's happening all nicely. To create this cell wall division, you have what is called a cell plate. So that basically begins to grow. And you can see they're basically just these structures that will give rise to this cell plate. And then you've got some other stuff going on. Again, you don't need to know this too in depth, but you just have to be aware that an animal cell doesn't have a cell wall, so you just have to split and you're fine. With cytokinesis in a plant cell, you have that extra step of forming that cell wall. So yeah, you can have a read for that process in a little bit more depth, but it's not too important to know all these kind of keywords and things like that. It's just important to understand the difference, as you can see in these diagrams ultimately. Hopefully that makes sense. And remember, this is kind of separate to mitosis. Like, I know there's a little bit of, I don't want to say debate, but like sometimes it's very easy to confuse cytokinesis as part of mitosis. Just understand that it is separate. Okay, so compared to mitosis, the way that prokaryotes divide, or for example, bacteria, binary fission, it's so simple. They literally just replicate their DNA and cut in half. So it's almost like the telophase part of mitosis, because you don't have to account for this like separation of like this intricate separation of your chromosomes and stuff like that, because in our prokaryotes there is one circular chromosome. So as you can see here, kind of this black squiggle represents that chromosome. The blue one is that now replicated. Again, they just go to either side of the cell splits in half, you have two cells. Very simple process. It's not as complex as mitosis, and it happens much, much faster. That's why we think of like when you think of bacteria, like what you guys know about bacteria, they replicate really rapidly, and it's super simple because it's such a quick, efficient process, really. So this can happen really, really rapidly. You're just doubling that genetic material and pinching off in the middle. That's ultimately it. But just be aware, things to be aware of. The idea that you have one circular chromosome. You have plasmids. These are just kind of other little offcuts of DNA, I would say. You talk about it a little bit more in 3-4. And then importantly, you have ribosomes. Remember that ribosomes, the difference between prokaryotes and eukaryotes have membrane-bound organelles. Ribosomes are not bound by a membrane. So that's kind of why they're in prokaryotes. Okay, so that was kind of talking about mitosis, I guess going backwards or technically forwards, like looking at the other parts of the cell. So we have our G1, which you talked about, it's literally just growing. S, you have the chromosomes duplicating. G2, growing a little bit more, preparing for mitosis. G0, this is something else. This is like a resting state. So we'll talk about here. The eukaryotic cell cycle is very complex, as you know. There are a lot of things that can go wrong, and things are generally bound to go wrong sometimes. So it's really important that there are checkpoints, because obviously if you have a cell that has uncontrollable growth, or if something's gone wrong with the synthesis of the DNA and that cell continues to divide, there can be other consequences further on. So these checkpoints are really important. So we have our G1 checkpoint. So after growing, it's just checking for these kind of stuff to make sure that nothing's too wrong there. You've then got your DNA synthesis. You then grow a little bit more. We're now at our G2 checkpoint, where the cell is checking, okay, did the DNA replication go okay? What is the cell looking like? Is it big enough for division? And then we have a checkpoint in the middle of mitosis as well at metaphase, and that is checking to make sure that those spindle fibers that we talked about, that they are attached properly to the chromosomes. Because again, if you pull too much of a chromosome or not enough of a chromosome, you can get that kind of imbalance in genetic information in your cells. So hopefully that makes sense. And if there were anything to go wrong at these checkpoints, the first kind of call of action is repair it. So if you have DNA damage or something like that, the cell will try to repair it. It'll repair it and then it will go on with the cell cycle. If the damage is irreparable, the cell will basically be programmed to die. So that's where we come into apoptosis. So apoptosis is, you've probably heard this term a lot, programmed cell death. Glossary term, remember that, super key. So in general, know that apoptosis, again, can be induced by different things, but it is very ordered, it's very programmed, and it's a very clean and efficient way of cells dying. It's a very necessary process as well. It's not always a bad thing. Sometimes it's necessary. It's used as a process as well to control for cell growth. So to make sure that there's not too many cells in the body. When you have old cells, to get rid of them. Cells that aren't serving a function anymore, getting rid of them as well. But then also kind of what we've talking about, like if there is DNA damage that is irreparable, then we'll have apoptosis usually as well. So in general, again, I'm not going to go too in depth, but you have your intrinsic, so think about the term, and your extrinsic. So think of intrinsic coming from inside, so can cause by like cell damage or damage to DNA, things like that. And it's caused by basically this molecule from the mitochondria, so called cytochrome C. Again, probably not too key to remember all these details, but just understand that it comes from within the cell. And so once this little molecule is released, we've got all of this kind of thing, and it leads to apoptosis. On the other hand, with extrinsic, it comes from outside the cell. So we have this little molecule, and so this is the cell here, and it comes from externally basically, and it signals the cell again to program and have that apoptosis, because that can be caused by extracellular things, such as infection, for example. As you can see here, again, you don't need to know the details of this, but if you see the term caspases, you can see that it's quite a similar process that results in ultimately the same thing. So while the initial thing may be different, the end result is the same. So we're looking at this idea of caspases. They are really important. I really highly recommend when you start to write your short answer questions and you're answering stuff with apoptosis, remember the term caspases. They're very central to this whole kind of process. Basically what they do is they initiate apoptosis by breaking everything down, like chopping everything up. So the cytoskeleton is what gives the cell its shape, its structure. So they chop that up. So we're getting this, like you can see here, this compared to this. We're losing our structure. We're cutting down our proteins, our organelles, things like that. So we're getting the cell shrinking, and what we see here, it starts to form blebs. So these little like offcuts, I don't know, like these little extensions of the cell. They basically break up, as we can see here, and then they get digested very nicely by our little phagocytes. So this stuff, again, I may be introducing some foreign words here. You'll learn about them, it'll make more sense. But ultimately we have the cell here. We lose all our structure, everything gets chopped up, we're forming these little blebs. Ultimately, these blebs actually branch off. So you can see how it's very neat. We're forming these little compartments that have all these like dead material in it, really. And then our little phagocyte, which is a type of immune cell here, gathers them all up and gets rid of them. And so you can see it prevents inflammation. So when you have your cells dying, you don't know, right? Like you're not seeing this really gruesome process where you're getting inflammation, where you're getting pain, anything like that. It's very neat. It's very efficient. So that's why it's that idea of programmed cell death. It's very programmed, very clinical almost. Okay, hopefully that makes sense. I know that was a lot of terms, so hopefully that's all okay. But again, pop anything in the Q&A if you have to. So here we have a ranking poll. So move the phases around so you can rank the steps of mitosis in the correct order. But yeah, hopefully that's making sense. Any questions, pop them in the Q&A and we'll get to them. Perfect, give you a couple of seconds more. Okay, lovely. That's looking pretty good. I know the answering system is a little bit different, so hopefully yours looks like what I've got up on the screen here. But remember that acronym. Acronyms, other memory aids, mnemonics, whatever it is, really, really helpful for bio, especially when there's a lot of content. I think I saw a question about study tips and stuff like that. Yeah, try to think about little memory aids that can help. And again, trying to make connections when you can. And it can really help get things in your brain. So with processes, when you have a lot of steps, yeah, remembering acronyms and stuff like that is really important. So PMAT, prophase, metaphase, anaphase, telophase. And obviously try not to, I mean, you can rote learn them, but try to think, M for middle or A for apart, stuff like that. That can help as well. But just remember the order and obviously what is involved with them. Okay, I think that was it for the cell cycle stuff. Oh, okay, never mind. Don't worry, we've got a little bit more. So this is when we're thinking about, okay, so what we were talking about before, this is kind of all the neat stuff, as I mentioned. So when stuff goes right, when stuff goes wrong. So when something goes wrong in the cell cycle and you have apoptosis, that's exactly what you want to happen. The cell dies, everything's good, nothing kind of negative is going on. However, sometimes we can get issues with the process of mitosis, which we discussed before, and it can lead to these things called neoplasms, which are basically just these unwanted growths, you could say. So they can be described as benign, potentially malignant, or malignant. So this is when we're starting to get into things such as cancer. So benign basically just means not harmful in this kind of context. So we're thinking of masses that are localized. So they're just sitting there. The excess mitosis, it's not kind of branching out. It's not affecting anything else, any major organs, anything like that. It's benign, right? It's pretty limited to where it is right now. Potentially malignant, that's kind of our step upwards. So that's when we're thinking about, at the moment, it is relatively benign. It's still localized, but it has the capacity to invade other tissues and become cancer. So that's why potentially malignant. Once it is malignant, that's what we're thinking of tissue invasion. So we have this excess mitosis and it's becoming harmful. It's basically spreading into areas where we don't want it to be. Okay, so in terms of this idea of what can lead to malignancy and even metastasis as well regarding cancer, we have these things called oncogenes and tumor suppressor genes. Again, I'll try to cover these as basically as I can. Don't be alarmed if you're not grasping it. Again, there is more to it and it'll probably make sense once you learn it in its completion. But oncogenes, basically, they have to be activated, whereas our tumor suppressor genes, as you can see here, like the arrow kind of means like, yes, like activate it. And this little thing means block it. So maybe I'll talk about our tumor suppressor genes first. So our tumor suppressors, as the name suggests, they suppress tumors. So you may have heard this one called P53. It's probably the most like famous talked about one, whatever it is, because it's the one that is often dodgy when we think about cancer. So P53, its role is, when we looked at that checkpoint before, if you remember that diagram, that'll suppress tumors by when there is kind of damage to the cell cycle. It's important in stopping it. So when we've got issues with mitosis or when we've got issues with DNA damage, cell growth, whatever it may be, the tumor suppressor genes job, or the tumor suppressor, the tumor suppressor's job is to stop the cell, right? It's to say, okay, something's gone wrong. Let's stop the cell. Let's, maybe make it to apoptosis. Let's try and fix it, whatever it is. With certain cancers, we see that the tumor suppressor, such as P53, it basically becomes faulty so that when there is an issue with the cell, we don't have anything saying stop it. So you've got DNA damage or you've got an issue with the growth of the cell. There's nothing there saying, okay, let's pause, let's stop the cell from growing. And that DNA damage or whatever it is that's gone wrong, that continues to be in that cell, and that cell will divide. And then that's when you've got issues, because then maybe you've got excess mitosis or you've got a mutation that can lead to cancer. On the other hand, we can also have our oncogenes. So our oncogenes are basically these genes that are capable of leading to cancer ultimately. And you have what are called proto-oncogenes. Again, I don't want to confuse you too much, but proto-oncogenes, they are these genes that just hang around And to basically be negative and to lead to cancer, they have to be activated and become oncogenes. If you think of like oncologist, it's someone that specializes in cancer, right? So an oncogene, it's kind of this cancer gene. So if you have a proto-oncogene, it's not doing anything, but once that proto-oncogene, perhaps something is mutated in that gene, something goes wrong, and that gene is now activated, it becomes an oncogene, and that can lead to cancer. So can you see why there's like pluses here and minuses here? Because when a tumor suppressor gene stops working, so we've got it kind of being knocked out, that's when we're leading to cancer. And here, when we get our oncogenes activated, that's when they can lead to cancer. So the way that I think of it is tumor suppressors I think of as the brakes, oncogenes I think of as the accelerators for like a cancer car, if you kind of think about it. So your brakes aren't working, that car is going to drive straight forward. You're pushing the accelerator with your oncogenes, the car is going to drive straight forward, and that can lead to cancer. Some of the traits we're thinking of is uncontrolled cell growth. So we're just thinking of heaps and heaps and heaps of mitosis that cannot be stopped, basically. And that apoptosis that we really would desperately need in a situation, it's not happening. And then that's when we can get other things such as cells invading tissues, organs, spreading of cancer through the bloodstream, stuff like that. Okay, in terms of mutagens, we'll talk about them in the next slide, but mutagens are some of the things you would have seen this in a lot of health campaigns. I'm sure you guys will be aware of some of them. So if we're thinking about UV radiation, why it's important to wear sunscreen, when we think of like melanoma, stuff like that. Cigarette smoke, so obviously we've heard that a lot in like lung cancer, things like that. Other things such as, processed meats, when you barbecue something and it goes like really black, that can be carcinogenic as well. And certain infectious agents, so we've got HPV, H. pylori as well. They can cause infections that can lead to cancer too. Okay, so in terms of the actual definition of our mutagens, so basically factors that cause mutations in DNA. Again, think of the words mutagens, so they're leading to mutations. Mitogens lead to mitosis, so just be aware of that. With bio, it's not a huge thing to get your spelling wrong, especially because you guys are in 1-2, your teacher is the one who'll be marking it, so I'm sure they'll be relatively lenient. With VCAR, like once you get into 3-4 and your exams, the rule is that you can make spelling mistakes, it's fine, as long as they're not basically other biological terms. So if you were to write mutagens and you wrote mitogens, like even though you're thinking of mutagens, because mitogens are an actual biological term, that spelling mistake wouldn't be accepted. So hopefully that makes sense. Like if you were to write like mutagens with 2 T's, like they know what you meant, you know what I mean? Like mutagens aren't another term. So just be careful with things like this, like mitogens and mutagens, they sound the same. They're like within the same realm of like cell division and stuff like that. So just be really careful. Carcinogens, basically what we were talking about before, those tumor suppressor genes, those proto-oncogenes, they're kind of more directly affecting them. And so that's why they're really leading to that cancer. So obviously mutagens, stuff we talked about before, they cause mutations in DNA, and those mutations can lead to cancer. But sometimes when we're thinking of carcinogens, those are like specific mutagens that cause cancer. Hopefully that kind of makes sense. So just some of the stuff we talked about before in terms of chemical things, physical things, biological things as well. Again, I'm sure these examples, you guys would be able to think of some of them off the top of your head based on like public health campaigns and stuff like that. Okay, so there are some other factors that may lead to, or I guess, aid the process of cancer developing. So loss of immunity. So if you are an immunocompromised individual, so basically your immune system isn't working as well as it should be, then it's not as strong perhaps the cells, there aren't as many immune cells as there should be. This may be more likely to lead to cancer, because in the event that perhaps these neoplasms, you have these unwanted growths, you have these genes kind of going on, your body may be able to destroy them. However, if your immune system is weaker, it will probably be less likely to. So that's why we can see that as well. And these are just some general things to be aware of. Again, I'm not going to go into them in detail, but think of metastasis, like spreading, evading growth suppressors. So we're, evading apoptosis as well. So you want to kill the cell. Cancer cells can be very tricky, and they can basically escape the process of apoptosis. and you can't turn off the excessive mitosis basically as well. So that's what we can kind of see here. Okay, so we're on our last bit of cell stuff now. So looking at stem cells, again, you guys may be familiar with this, but stem cells are basically like cells that are a blank canvas. So they haven't differentiated into a specialized cell yet. So my teacher used to say like, I don't know, you can kind of think of your cells as stem cells. You haven't specialized into a uni course or a TAFE course or whatever vocation, stuff like that. And you might think of like babies as really basic stem cells. Then you go into primary school, maybe you're a little bit more specialized. Then you go into high school, you're choosing a VCE subjects, you're a little bit more specialized. You go to whatever you do after uni, then you go into a job, after school, then whatever you do to get to a job. And then by the time you're an old person, you're very specialized. So I don't know. That's how I sometimes think of stem cells as well. Basically, this idea of they come a little bit more specialized and a bit more specialized and a bit more specialized. So we can see that here. So obviously these are like all examples of different cell types, and they all come from the same basic one. So if we're thinking of in an embryo, so thinking of like the really basic like fertilizations just kind of happened. Obviously the cells that you have in there can become anything. You then have some other cells that are pluripotent. So you have totipotent or totipotent. I don't know, you hear it called some in different ways. But basically it can become anything. So I think of like toti, think of like total. Pluripotent basically means it can become most stuff. So we have here like endoderm, mesoderm, ectoderm. So these are kind of technical terms, but basically when you're in the like as an embryo, there are these three different types. And the reason why these are pluripotent and not totipotent, totipotent, they can become ultimately anything, but these cells cannot become like each other or that basic embryonic stem cell. So that's why it's not totipotent, if that makes sense. But they basically can become anything. Then they become a little bit more specialized, and then we call them multipotent. So you can still become some certain types of cells, but just not everything. So if you're thinking about bone marrow, skin, cord blood, whatever, stuff like that, they can become different things. So for example, bone marrow, so you can become a red blood cell, you can become a heart muscle, so like a cardiomyocyte basically. You can become different cells, but see how they're within kind of a similar realm. But these cells can't become a lung cell or they can't become a skin cell. So that's why we're a little bit more specialized. And once we are specialized cells, so once we're a red blood cell, we're not becoming anything else. So you're completely specialized there. And that's what we'd call unipotent. So think of uni as one. You can only be one type of cell. So like a skin cell or a neuron, you're only dividing to become that exact same type of cell. You're not dividing to become a different type of cell. Hopefully that makes sense. You can just see that kind of level of specialization. So hopefully that analogy of like a primary school child is kind of making sense. Okay, in general, we can see here our embryonic stem cells. So remember this idea of endoderm, mesoderm, ectoderm. They're just parts of the embryo really that can give rise to different things. So you can see how they're totipotent or pluripotent. So they can become a lot of different things. And they can replicate indefinitely. So basically they can replicate and replicate and replicate and replicate. So if you were to, I guess, like harvest those stem cells, they can become anything and they can replicate continuously. If you were to get an adult stem cell, they are multipotent or unipotent. So thinking a little bit down the line, you're a little bit more specialized, yeah? So you can use these stem cells to repair damaged tissue, stuff like that. However, these cells cannot replicate indefinitely. They still have a finite time because they are from an adult stem cell. That is the basic gist of it. Obviously, there are ethical things associated with this. So you can see that there's pros and cons of using embryonic stem cells and adult stem cells, but questions are raised when it comes into, harvesting embryonic stem cells or harvesting adult stem cells as well, kind of reversing them, things like that. But yeah, that's kind of a basic gist of stem cells there. So we'll have another Slido poll. So what are some examples of mutagens? So this is a word cloud, so you can pop in, if you remember, some examples of mutagens. Any clue? So we talked about different classes, like we mentioned, like there's like biological, physical, chemical. Yeah, so everyone's kind of thinking the same thing with radiation. Yeah. Yeah, definitely. And I guess that's something that always comes to mind in summer, like around this time of the year, um, in terms of, what am I saying? Sunscreen, stuff like that. Yeah, cigarettes. So you see that on cigarette packets and stuff like that as well. So we're thinking obviously associated with different types of cancer, but obviously UV radiation in the sun, we're thinking mostly with like skin cancer, melanoma. Cigarettes, we're thinking more so with lung cancer, stuff like that. But obviously there can be other stuff. Yeah. Nitrates, HPV. Yep. So remember we've got our like microorganisms, so they can cause infection. Yep. Skincare. Yep. Burnt meats. Yep. Good. Okay. Perfect. I feel like that's most of them. So yeah, we talked about a couple of them before. Okay, let me lock that one now. Okay, so kind of moving on now to our second block of content, we will go through the plant systems, the animal systems, and homeostasis like we discussed at the start. And then again, we'll have the same sort of thing, so with our Slido polls and our questions at the end. So as mentioned, I'll try to get through this one fairly quickly. Not too quickly, but enough so that we do have time at the end. So basically with our plant systems and our animal systems, again, think big picture. I'm kind of talking to you guys about, this idea of vascular bundles, and we're going from specialized cell to tissue to organ to organism. So that is a big picture. But think of the even bigger picture. On the study design, we're talking about plant systems and we're talking about animal systems. So they're just this idea of when you have, almost like stem cells, like that kind of hierarchy or those levels to organization that work to power this bigger thing. Basically, that's what we're looking at. And we're looking at an example of our plant system here, and we look at multiple examples of animal systems. And it's just this idea of how we are able to have a plant that functions, how we're able to have the human body that functions with stuff like the digestive system or the endocrine system, for example, and how these levels of organization, rather than like one big clump of a system, how that kind of helps the organism function pretty efficiently. So we have this idea, these general levels of organism, and this applies to all of them within plants and animals. So we have our specialized cells. That's kind of our most basic little unit. We have then a bunch of these cells together that form a tissue. A bunch of these tissues together forms our organ. A bunch of these organs together forms our system. A bunch of these systems together forms our organism. So hopefully that's kind of making sense. In this specific example, our specialized cell, we've got our root hair cells. So we're thinking at the root of the plant, they form tissue, they form roots. So that's kind of making sense. And then this is kind of the important part. This is an image that you'll probably get used to, a diagram as well, of these little vascular bundles. So a bunch of these vascular bundles together make up the vascular system of the plant. And then obviously that leads to the entire organism. So talking about these a little bit more specifically, they have non-vascular plants as well. So maybe I'll just address these. These are a little bit more simple and they just work through osmosis. So the process we talked about before. Remember, we're thinking about water moving from high to low, and that's just how stuff kind of gets around. Vascular plants are a little bit more complex, and we're thinking about an actual more established system. So we need to move water through the plant. We also need to move minerals. We need to move products from photosynthesis, so particularly like glucose, sugars, where we get our energy. I will sort of flag this as well. I know you don't look into photosynthesis and cellular respiration in detail that much in Bio 1-2 anymore. However, it's a really important thing. Like when we were looking at mitochondria and chloroplasts, I don't want you to think photosynthesis, plants, cellular respiration, animals, and that's that. Understand, obviously, photosynthesis is only plants, but cellular respiration is both of them. We all, all the organisms need to get energy somehow. So plants also have cellular respiration. They also need glucose. try not to fall into that trap of thinking only animals have cellular respiration, only plants have photosynthesis. Remember that plants do need ATP as well. So that's why it's important to move these photosynthetic products. And the way we do that, focusing on these vascular bundles, we're looking at this idea of xylem and phloem. And that's kind of what you saw, like they look like these little circle bundles. So we'll kind of talk about that a bit more. So in terms of our transport, as you can see with this like transection, I guess this like little view that we have, if this is like a stem or whatever, I don't know, they go vertically. So we have this kind of transport system. You can think of it moving vertically through the plant because obviously we need to get from the roots to the leaves and back as well. So we have xylem, phloem, lignin as well. It's a bit less important. Lignin is basically just to provide structure to the plant. Xylem is really important in this transport of water and nutrients from the bottom up. So we're thinking from our roots, drawing that up to the leaves. Our phloem is really important, particularly from getting our sugars, so that are produced in photosynthesis, so our glucose, stuff like that, from the leaves where the photosynthesis happens to the roots, as the roots really need that energy to grow. Okay, so we can see these in our diagrams here. So you can see the lignin is just kind of what you'd kind of expect a structural kind of molecule to look like. So it just gives it that structure. You've got your xylem here and your phloem, and again, part of those vascular bundles. Here are some differences. So xylem is one-way movement. So we're thinking of water and minerals drawing upwards. With phloem, we can see kind of the movement of our organic molecules. So when you see organic molecules, just kind of think sugars, basically. And those can move up and down. With our xylem as well, the outer cells are dead. So they're not living. And that can kind of make it a little bit more rigid as well. And that just enables the water to draw through. We can see it's one way, which we discussed. And there's no kind of sieve plate that we can see here. So here, almost like, again, beware of drawing comparisons because I don't want to get you like too confused. But if you think of like valves in veins, you can almost kind of think about it that way. But this idea that the water just like passes through, there's no separation between these cells. However, with this one, we do have that separation. So those are some kind of major differences to be aware of. Okay, so in terms of the process, so transpiration, here we're thinking of xylem especially, and we're thinking of water. So as you can see here, water is moving upwards. So remember, we're thinking from the roots to the leaves. So it's basically being like it's just, think of it like a vacuum. Like I just think of this like something being like drawn up really. So through the root hair cells, so those specialized cells that we discussed, they're going up and up, and so they're moving through the plant. And we're using this process particularly of cohesion. So there's adhesion as well. But cohesion is basically like when you guys will be familiar with this, like when water, like, if you have two drops of water and then they come together and they form one big one, it's kind of like that. So it's this idea of they almost pull each other along, these little water droplets, and it moves its way up. And remember, we're thinking of osmosis as well and kind of things like that too, where it's moving from a low concentration to a high concentration, but we're getting this ability to be pulled up via transpiration. So we're thinking of this adhesion is just kind of like sticking to the cell wall and moving up. Cohesion is this idea of one water molecule kind of linking to another. Like think of the words like cohesion, working like cohesively, and moving upward, and you basically just get this drawing up of water. Hopefully that makes sense. And again, think of that with the xylem in particular. So something else important with kind of water is this idea of stomata. So you can see here, this is open, and this is closed. And so the stomata is the actual sort of holes in the plants, and the things that open and close them are guard cells. So we're getting this idea of water going up, 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 up. So once we get to like, the peak, the leaves of the plant, water can exit that way basically. So this is an example, maybe I'll talk through it first. So if you have your guard cells, remember that these are the things that cause the stomata to open and close. If the guard cells are full, as we can see here, so there's a lot of water, they're basically full, they're being pushed almost, and they push open. And that makes sense because you've got a lot of water, enough water to push it out. That means there's a lot of water and therefore you can let some go. On the other hand, if the guard cells are flaccid, so they're a little bit more loose, they're not as turgid and they're not pushing things out. Therefore, you don't have that much water, so you don't have enough to push them open. But again, that makes sense because you want to keep the stomata closed because you don't have that much water, so you don't want the water to evaporate. See how that kind of makes sense? This is only just like one example of this type of thing in bio. Like you'll see a lot more throughout the year. You'll see heaps of it in year 12. This idea of when the actual thing, I guess that's affected. So if we're thinking about like transpiration or evaporation here, is the actual thing that causes, I'm explaining this really badly, like the actual thing that causes that kind of process to occur or to not. So if you have not enough water, you don't have enough water to fill the guard cells to open them. Therefore, you're not going to let the water out. You see what I mean in that way? Yeah, you'll learn about that a lot more. It's kind of this feedback system where the actual thing, we'll almost talk about it a little bit in homeostasis, where it's like the actual thing itself can be the thing that determines the pathway of that thing. I know that's such a general way to explain it, but you see it a lot more like in genetics and like protein production and stuff like that next year. Okay, so some factors that affect transpiration rate. So we've got humidity, temperature, wind, and the time as well. So again, with these, I know it seems like a lot, but just think of it in the relationship to water and it should make sense. So if it's humid, transpiration will decrease because there's a lot of water vapor in the air. So remember that if it's humid, it's hot, but the air is muggy. There's a lot of water in the air. So therefore, you basically have, there's less of a water concentration. So there's not, when we think about movement, like high concentration to low concentration, if there's lots of water in the air, that means the air outside and kind of the leaves inside, it's a pretty similar concentration. So less water is going to want to come out of the plant, if that makes sense. Like you can almost think of it like isotonic. So that's the idea there. If it's humidity, you're thinking more water in the air. There's no reason why water will want to leave the plant. So therefore you've got transpiration decreasing. You have less of it kind of pulling up the plant and coming out. Temperature. So as temperature increases, as it gets hotter, so we're not thinking of humid hot, but we're thinking of just as the temperature increases, you're going to have transpiration increasing because think of evaporation. Wind. So as wind blows, we're getting transpiration increasing. Again, evaporation. So if you've got more force of the wind, if that kind of makes sense, those water molecules are going to evaporate. If it's hotter, water molecules are going to evaporate. And time of day, again, you can kind of think about it tying into the first two. So at night, it's going to be cooler. So temperature lower means transpiration rate lower. And also it's more humid at night than it is in the day. So we've got transpiration decreasing. And then when the sun comes out and it's a bit warmer, it's a bit less humid, you have that evaporation. So hopefully that kind of makes sense. Okay, so now when we're thinking about the phloem kind of tying into this, we're looking at translocation. So this is a little bit more complex. So basically you've got our transpiration. So we're moving upright. And again, we're thinking from a high region of water concentration to a region of low water concentration. So we'll follow the steps here. So we have our photosynthesis. We've got our glucose. Here it's sucrose. Just think of a sugar, right? It doesn't really matter about the names. So we've got our sugar, and this is coming into our cells. I'm not going to talk about the cells too much, but just think from our leaves to our phloem, right? We're moving this sugar along. So now there's a lot of sugar here. What is going to happen, again, think of osmosis. We have a lot of water here and a lot of sugar here. Hopefully this is sounding a little bit familiar. Think of the diagram we saw really early on with the osmosis process. There's more sugar over here. Water is going to flow into the phloem. So we've got water coming along here. This is going to cause this area of the phloem to swell up. We've got a lot of water, right? It's growing. And that osmotic pressure, so the pressure of the water, is going to push everything down. So hopefully that's making sense. We've got a lot of sugar. Water's going to come in because of osmosis. That's going to swell up. It's going to create this huge kind of pressure and push everything down. What's going to go with it? The sugar. So the sugar is coming down, 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 down. And that's where we're getting this translocation of sucrose. And it's going to these areas of the plant, such as the root cell. So where photosynthesis doesn't really happen, we're thinking of like roots. And that's putting the sugar and kind of the molecules that we need in there. So you can see that unloading. And then now there's not that much sugar there, the water will kind of go back. So you can see how it's this nice cycle. Like, I know I'm just giving a general overview, but you can see that the processes really tie into each other and it just creates this very well-oiled machine. And you'll see that with all the other systems as well. So kind of, again, think about it bigger picture. What's the general objective? And we're just wanting to get water from areas where there's a lot of it to areas where there's less. And same thing with the sugar, areas where we've kind of collected it, where there's a lot of it, to areas where there's less. And we're using kind of both of these systems to allow that to happen. So hopefully that kind of makes sense too. Okay, so we'll do a quick Slido poll now. Okay, so transpiration rate increases when what? We'll try to be as quick as we can with these ones so we can get through and answer some questions. Yeah, looking good. Yeah, perfect. So essentially it is when our wind increases. So I'll lock that one here. Again, sorry if you're not getting time in, hopefully it's enough time. But yeah, when our wind increases, remember thinking of that movement of the wind, it's going to evaporate that water. Like think of like a hairdryer, maybe. Like obviously the heat, so when temperature increases, we're increasing the rate of that water evaporation from your head. But like even when your hairdryer is on the cool setting, it can still dry your hair from that wind pressure, I guess you could say, or that movement. So that's kind of what you can think of. Remember when there's a lot of water vapor in the air, that means when it's humid, think about that there's a lot of water in the air and almost there's a lot of water in the plant as well. It's the same. Water isn't going to want to come out. So you can kind of think about like osmosis principles and stuff there. Yeah. Okay. So we'll now move to the animal system. Hopefully that makes sense. Again, these are just general overviews. Pop anything in Q&A. So digestive system, we've got the general kind of movement of it here. So we have all of these different parts that you may be familiar with. So generally we're going from the mouth, we're moving the food from the mouth to the stomach via the esophagus. We've got input from the liver, from the pancreas. Our food moves from the stomach through to the small intestine, then through to the large intestine until it's excreted out as feces. Okay, so in terms of the purpose, basically we get our nutrients from our food, we get our energy. Again, with cellular respiration, we need that source of glucose to provide ATP. Food is where we get it from. So obviously we need glucose in its like raw form. In food, that's not how it's presented. So we have to digest and break down the food in order to get the glucose, basically. So digestion occurs both to physical and chemical mechanisms. So we've got physical stuff like peristalsis. So when the esophagus actually, like you can kind of see it in that video. Sorry, I like pointed at it. You can kind of see it here. It moves. So that idea of peristaltic movement, it's that actual like constricting and relaxing of your esophagus. Your intestines do it as well, and it actually physically moves the food along. But then think about, the acid in your stomach, the other digestive enzymes. That's the chemical part of digestion as well. So yeah, essentially, yeah, we just have to get our nutrients from food, basically, and that's why the digestive system is really, really important. So very similar to the kind of flow we saw with the vascular bundles and the plant. So for our digestive system, the specialized cell, this is kind of just an example. There are obviously different cells in the stomach and the esophagus and all this stuff. But with the small intestine, we're thinking of villi. So you guys may be familiar with this. It's like these little extensions that are in your small intestine and they absorb nutrients and things like that from food. Super, super important. So all of them together, they create the tissue, and then that creates your small intestine, which is just one organ in your digestive system, leading to humans as well. So you can see this, again, this step-by-step kind of organization there. Okay, looking at the excretory system. So we have kidneys. Again, this is quite a complicated diagram, I would say. The excretory system for me is probably the hardest one to grasp initially, I would say, just because there's a lot of different terms and you have to think about it. It's a very process-based kind of thing. But yeah, just as a general overview, we have our kidneys here and we have these little specialized cells, so our nephrons. And we can see, as you can see, this is what a nephron looks like. It's made up of these other things here. So we've got our important things, probably like these two, proximal convoluted tubule and your distal convoluted tubule. Again, I'm just mentioning the names now, but what they're really important for is basically absorbing things that your body needs and excreting things that they don't. So the kidneys are super important in filtering. So they filter your blood and they will draw out things, again, that the body needs more of, and anything that can go to waste, that will be your urine essentially, and you'll excrete that. So yeah, you'll learn more about that in an in-depth process of that, but hopefully this kind of familiarizes you a little bit with what nephrons look like. So we can see this is a bit of a more basic drawing of this. So here, this is what's important. So Bowman's capsule, you can see that this is the blood and this is the part where we're actually doing the filtering. So blood basically enters your kidney. Again, like literally just think of your kidney as like this little filtering system. So we have a Bowman's capsule here, and this will filter out basically anything that can be excreted really. So then everything else, the blood just continues on flowing. So once we've kind of excreted things here, this will go through all of this stuff. So your proximal convoluted tubule, your distal convoluted tubule, it goes through all this. An important part here is your loop of Henle. I don't think it's written here, but here, your loop of Henle. This is really important in basically absorbing water. So if you are a little bit dehydrated, like you can absorb some more water from the urine, and that's when you'll produce like really concentrated urine. Whereas, if you're getting a lot of water in, maybe like your urine can pass and you won't absorb as much water because you're getting enough in, if not even more than you need. So that's a really important part as well. So you can see here we're absorbing things, salts, glucose, amino acids, water, stuff like that. Again, that can be absorbed out, and everything that is not reabsorbed, so everything that's not kind of taken in by all of these, that gets shoved out basically as your urine. So hopefully that makes sense. So here you can see the step of filtering. Again, really important, think of this idea of filtering. Here you reabsorb stuff. 3, don't worry about it too much, but 4, you're basically just pushing out waste. So anything that doesn't get reabsorbed, essentially. Hopefully that kind of makes sense too. Again, it is a relatively complex process, but just understand it's this idea of excreting substances that your body doesn't really need anymore. So in terms of our specialization here, we've got our nephrons, we've got our kidney tissue leading to our organ of the kidney, forms our excretory system, also known as our urinary system as well, leading to us as the organism. Okay, last one we look at is the endocrine system. So we've got our glands, so those are the organs we're kind of referring to here. They are found everywhere. I'm sure, as you know, endocrine system, I think you do from memory, unless they change it. You touched on it in year 10. Here you look at it in a little bit more detail. So you've got your endocrine glands, and they're really important at producing specific hormones. And with hormones, we think about them being these sort of chemical messengers that are relatively slow acting. So they have these kind of diffuse effects most of the time. And those can be things like growth hormone. They can control, if you think about insulin, like things with glucose, even hunger, sleep hormone, stuff like that as well. Like they have a lot of different, I guess, applications and uses, obviously depending on where they're released, when they're released as well, stuff like that. A really important thing to focus on is this idea that they do travel in the bloodstream. So therefore they can reach things that are far away, but however, they may take time. They may be a little bit slow. Okay, so in terms of glands, we're thinking about controlling how much of each hormone is released. And this depends on what is triggering the hormone release and how much of the hormone is already there. So we can see, you would have had this, like, things affecting hormones. So you can see changes in the fluid balance. You can see infections as well, stuff like stress, age as well. Obviously that differs in terms of hormone release too. Okay, so obviously this is quite a general overview. Like we'll look at homeostasis a little bit and you can kind of think about it. But wherever the glands are, they'll have these special cells, so your epithelial cells, and they're secretory, so they secrete something. So basically, like we talked about organelles before, they'll produce, for example, let's say it's a protein hormone. They'll produce these proteins that are hormones, and then they'll export them, so they'll secrete them from the cell. And those will be our glands, and these glands form our endocrine system. Okay, we'll look at a Slido poll quickly. So this one should be okay, hopefully. Which of the following is not a specialized cell? Given what we were just talking about. So think about the different systems. Think about which of these is not a cell, but is something obviously higher up in the chain. Yeah. Okay, looking here again. Sorry to cut some of the answers off, but just in the sake of time. So yeah, gland is not a specialized cell. So our gland will be the organ. And within those glands, we'll have our cells, our secretory epithelial cells, which will secrete those hormones. So nephrons are the specialized cells within the kidneys, so the excretory system, and villi are the specialized cells within the small intestine. So thinking of digestive system there. Okay, I think we've only got a little bit left, so time should be okay. So looking at homeostasis, basically we've spoken a little bit. This is really important, obviously with a lot of different systems, but particularly endocrine. We're thinking of stimulus response model. So basically we get a stimulus, a receptor detects that essentially. We act upon that and then we initiate this response. So we can see here a stimulus, again, pop these in your glossary, know these words. So we're thinking a change in the environment. So something that will trigger something else, basically, trigger a change. A receptor, so a receptor detects this stimuli and then it converts us into, it says these electrical nerve impulses. If you're aware of what neurons are, we don't talk about them much today. But these neurons, they're basically, as you can see here, the central nervous system. So that's our brain and our spinal cord. So these neurons will detect things. So say temperature is too high. So there might be something in the skin. So this is kind of when we're thinking of particularly this response, but you will get something that detects that. So the hypothalamus will detect that, but there will be other things that kind of send it to the hypothalamus first. So perhaps it's your skin, maybe detecting that the temperature is too hot or something like that. So then that receptor will send that signal to the brain. So therefore the hypothalamus realizes, okay, it's too hot. What am I going to do? It has to initiate a response, and that response is to cool the body down, right? So we're going to act on the skin, the blood vessels, the cortex as well. So in terms of the brain, we're moving us, the body, to a shady spot. So the sun isn't shining down on us, so we're a little bit cooler. At the same time, our blood vessels will dilate, so we'll get kind of our red face as we try to lose some of that heat. And the skin as well will begin to sweat as we want, like when our sweat evaporates, like that liquid, that cools us down as well. So again, stimulus, our receptor that detects that, we're initiating this response through our effectors. So they produce a response to the stimulus. So hopefully that kind of makes sense. Again, I know it seems like a lot of words, but just think about the process logically. The temperature is too high, you want to cool yourself down. These are the three things that happen to initiate that. Okay, this is an interesting one. Take it as you will, but again, it's just this idea of pushing stimulus, receptor, response. It's important to think about neurons. I know we don't talk about neurons too much, but understand that they are really important in transmitting this message. So they're very important in detecting what's going on, sending that to the brain, and receiving that message from the brain and sending it to the effector, which might be, the blood vessels, or maybe it's the hand, stuff like that. Okay, so you can see, for example, we've got chemoreceptors. So you see, it's not just like general things, in the skin. It may be receiving the sense of pain, receiving temperature even pressure as well. Chemoreceptors, maybe it's sound, think of all your senses, right? Maybe it's vision, all that stuff. The neurons there will send that message to the brain. The brain will decide how to act on it. So you can see here, the brain sends a message. Afferent and efferent, don't worry about it too much. It just means like sending something to the brain versus from the brain. And then the response. So that's how it works. Just think of stimulus, receptor, message being kind of translated, and then response. Okay. Oh, I think my thing is frozen. Okay, bear with me. Okay, there it is. Hopefully that's okay. This is just showing you kind of what it looks like in terms of hormones in the endocrine. So we were talking about your secretory cells and stuff like that. So we have the blood in the capillaries, and I can't emphasize this enough. Remember that endocrine stuff travels in the bloodstream. So we're traveling from a gland that may be somewhere else. So it may be in the brain, and then it may have effects on, a muscle somewhere else. Or we might be sending something from the adrenal gland. So those sit on your kidneys, and we might be sending it to, let's say, the brain, or to the eyes, or to the heart, something like that as well. So they travel through the bloodstream. That's very important to be aware of. Okay, ways of controlling homeostasis. So these are the specific ones that are listed on the study design. So thermoregulation, we're thinking of temperature. Osmoregulation, we're thinking of water, blood glucose. So thermoregulation, we're thinking of temperature, as we mentioned, and it's kind of the example we looked at before. So when it gets too hot, we're thinking of sweating, we're thinking of vasodilation. When it gets too cold, we're thinking of shivering, we're thinking of vasoconstriction, stuff like that. And you see how it's just this idea of balancing. So obviously there's more to it, and you'll learn about the ins and outs of it a little bit more kind of in your course and in the year. But it's just this idea of keeping it balanced. So keeping homeostasis is basically just this idea, if you break it down, like maintaining the body's kind of right levels at all times. So there's a narrow range within which the body can operate and function at its best. And homeostasis is all about making sure that we don't venture out of these limits. So making sure that our body doesn't get too hot, making sure that our body doesn't get too cold, by controlling when we vasodilate, when we vasoconstrict, when we sweat, and when we shiver, stuff like that. It's all about keeping it within these narrow limits. Same thing applies with osmoregulation, not being too dehydrated, not being overhydrated perhaps. So it's this idea of the hormone involved is ADH, so antidiuretic hormone. Again, it's a little bit technical. Diuretic, think of like diuretics, they make you like pee essentially. So it's kind of what we talked about with the excretory system. It makes a lot more sense once you study the excretory system in depth, and then you have a look at osmoregulation. It ties it together so nicely. But with osmoregulation, we're thinking about if you are dehydrated, we're basically going to, like I mentioned with the lip of Henle and everything that goes on in the nephron, we're going to absorb as much water as we can, and we're going to release this anti-diuretic hormone. So anti-diuretic, we don't want to pee. That sort of makes sense. So you're going to excrete less water. Versus on the other hand, if your water level is kind of fine, then you won't have as much levels of antidiuretic hormone. You'll be able to pee as the body wants you to. And you're not absorbing or reabsorbing too much water because the body's got enough. Lastly, when we look at blood glucose, we're thinking about insulin here. So when we eat a meal, we've got a lot of glucose floating around in our bloodstream is when we release insulin. So insulin is the hormone. You'll go into this in more depth. So again, don't be concerned if you're not grasping all the hormones and stuff like that. I'm just speaking about it generally. But yeah, think you've eaten a meal, you've got a lot of sugar floating around in your body now. So we release insulin, and that brings the glucose into the cells. So your blood glucose in your bloodstream is high. The body detects that and goes, okay, time to release this insulin. Let's bring these cells in. Bring these glucose into the cells. On the other hand, if your blood glucose is low, the body detects that as well. And then you'll start to break down your stores of glucose. You'll start to release more glucose into the blood. so that the body has got more energy, basically, because you need enough glucose for ATP. It's really important. So that's why, again, with all three of these things, keeping the body within these narrow limits. Okay, hopefully that makes sense. I think this is one of the last things we cover. So we're looking at when homeostatic stuff basically goes wrong. So thyroxine, we're thinking of the thyroid. So the thyroid is just a little organ that sits kind of with your neck. So thyroxine is a hormone. And when there's overproduction of that, so basically when homeostasis goes a little bit wrong and we don't keep this under control, we can basically get this, this overproduction of thyroxine, and that can lead to hyperthyroidism essentially, and it can lead to what we see here as like this goiter. So it's like this really large mass on the neck that's caused by this enlarged thyroid. These two here represent insulin. So again, these are specific things that are specified on the study design. So we're thinking of type 1 diabetes. So this is when you have a lack of insulin. Basically, the body destroys the cells that produce insulin. So as you can see here, we've got our little sugar molecules, and this represents our insulin. So in a normal condition, we have our insulin, and this allows the glucose to come into the cell. With type 1 diabetes, we don't have that insulin production. Therefore, glucose is just kind of kept in the bloodstream, and there's always quite a high level of glucose. And obviously not getting glucose into the cells, that's a bad thing, because we can't then have our cellular respiration and produce our ATP. Something else that's specified under the study design is hypoglycemia. So we can see on the left here, basically we've got low blood sugar. That's what it means, like hypo, low, glycemia, so sugar in the blood. So here we have enough sugar in the blood basically, and assuming that goes into all of our cells. With hypoglycemia, we've basically not got enough, and that can sometimes happen, especially with diabetes medication. So with this, we can see that there's a high level of blood glucose in the blood. So sometimes we'll prescribe medication, obviously, to those people with diabetes, and that can cause actually the opposite effect, because the medicine almost works too well. And so that can cause hypoglycemia, which is what we can kind of see here. And that can lead to like dizziness and things that happen when you have too low of a blood sugar level. Okay, hopefully that makes sense. I think that